Approximately 1 in every 60 births results in the delivery of twins. DNA-based testing is the most accurate method to determine if twins are identical or fraternal. The medical term for this is twin zygosity testing. Twins are either dizygotic, commonly referred to as fraternal, or monozygotic, referred to as identical. 70% of twin births are dizygotic, while the remaining 30% are monozygotic. Dizygotic twins are the result of two different eggs being fertilized by two separate sperm and resulting in two completely distinct and separate pregnancies, which happen to be carried out in the womb at the same time. Dizygotic twins have the same random chance of inheriting genetic code from their parents as any other siblings. Like any sibling pair, on average, dizygotic twins share half of their genetic code. Monozygotic twins are the result of one egg fertilized by a single sperm. One pregnancy literally splits in two between the 4th and 12th day after conception. Although monozygotic twins indeed start with the same genetic blueprint, subsequent genetic and environmental forces may lead to subtle to dramatic differences in appearance. In extremely rare instances, one monozygotic twin can be a boy while the other is a girl. Many parents are given misinformation at the time of their twins' birth based on incomplete or erroneous examination of the placenta. Certainly, the number of placentas does not predict zygosity. 1/3 of monozygotic twins have two placentas, and 42% of dizygotic twins have a single fused placenta. Most often, zygosity testing is used to satisfy curiosity. However, there are medical reasons to definitively know zygosity. Because monozygotic twins have the same genetic code, they may be able to help each other in extreme medical circumstances. Usually, identical twins have the same blood type, so they could share blood in emergencies. In the event of organ transplantation, identical twins would have the best chance for success. Affiltated Genetics has performed more zygosity tests for multiples than any other laboratory offering this service. Since our inception in 1994, we have performed zygosity tests for many university studies, private genetics labs, pediatricians, and the public. Dr. Nancy Siegel, university professor and author of numerous books about twinning, says,
"When it comes to testing twins, I have relied upon Affiltated Genetics for years. The supportive staff, easy-to-use materials, and quick turnaround make doing research a real pleasure."
Affiltated Genetics is one of less than 50 laboratories worldwide that is accredited by the American Association of Blood Banks for relationship testing. Further accreditation status with CLIA and College of American Pathologists distinguish AGI from most other labs. Dr. Kenneth Ward, MD, is the founder and laboratory director of Affiltated Genetics. Dr. Ward is among the few physicians in the country who is board certified in 4 medical specialties: OBGYN, maternal fetal medicine, medical genetics, and molecular genetics. Before devoting his full-time efforts towards genetic research and testing in a private setting, Dr. Ward's academic accomplishments included former chair of the Department of Obstetrics and Gynecology and Women's Health at the University of Hawaii. As a professor of OBGYN and human genetics, Dr. Ward founded and ran the University of Utah's DNA Diagnostic Laboratory. He led the efforts to discover a number of human disease genes as well as genetic associations for several pregnancy-related diseases. As a perinatologist, Dr. Ward has personally cared for thousands of women having multiple births over his career. Once you order a zygosity test, a sample collection kit is mailed to your home via priority mail. The sample collection kit contains all materials needed for the collection of 2 individuals' DNA. The process is easy and non-intrusive. Cotton swabs are used to collect the cheek cells and saliva from inside the mouth. Step-by-step instructions are provided with the kit. Simply return the swab samples in the provided postage-paid packaging to Affiltated Genetics. Once the swabs are received in our lab, all samples are assigned a unique barcode. DNA is extracted from each sample, then through a process called PCR, 15 individual well-characterized markers, which have been selected for their diversity among human beings, are analyzed and the twins' genotypes are compared. A letter of results signed by Dr. Ward is usually mailed within 7 days from the date our laboratory receives the samples. Our testing is affordable, reliable, and highly accurate. Our cheek swab DNA testing gives 99.9% or greater accuracy. All results are kept completely confidential, and leftover DNA is destroyed 30 days after the test is completed. If you have any questions or wish to order a test, we encourage you to call our helpful customer service team at 1-800 888-362-5559 or visit our website at affiliatedgenetics.com and order your test through our secure ordering page.